Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Novartis, Allergan, Janssen, Biogen, AGTC, MeiraGTx, and the SickKids Foundation. We'd also like to thank Accessible Media Inc., our national accessibility partner. Everyone should download the AMI app, which is a fabulous resource, and you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoyed this webinar and share your thoughts with us in the comment section below.
Thank you for the opportunity to participate in this panel. My name is Hester van Diepen. I work at a small biotech company in Leiden in the Netherlands. It's called ProQR Therapeutics. And this company was founded about 5 years ago. And the aim of our company is to develop new drugs for inherited retinal diseases. And to develop these drugs, we make use of a specific technology. So we make use of the same technology to develop drugs for different diseases. And this is a RNA technology. So our technology targets the RNA, and the RNA is the blueprint or the message of the DNA. So it's transcribed from your genes. And then you get the RNA, which is then translated into proteins, which are important for many physiological functions as well as in your eye. So important to note here that our technology is different compared to gene therapy, because gene therapy targets the DNA, whereas our technology targets the RNA, and it's more, so it doesn't affect your genes, but it restores the mutation within the RNA. So we aim to develop drugs for different inherited retinal diseases. And so far we have 3 main programs which focus on, our main program is a drug called sepofarsen, which is a drug for Leber's congenital amaurosis. We have a drug in development for Usher syndrome, and then retinitis pigmentosa within Usher syndrome, and a third main program targeting autosomal dominant retinitis pigmentosa. So I am the project lead of the Usher program, so I would like to give this program today as an example of drug development. I joined ProQR about 4 years ago, so a little bit after the start of the company, as a scientist in the discovery department of the company. And I joined the team with the aim to find a new drug for retinitis pigmentosa within Usher syndrome. So this drug is now called QR421A, but we started by performing a screen in several test models to find the most effective drug. And an example of one of these models is the zebrafish. So we did this research in collaboration with Radboud University in Nijmegen in the Netherlands. And they developed a diseased model for Usher syndrome, a diseased zebrafish model. So we could use this model to test the effect of our drug and to test the hypothesis if our drug would possibly be effective in humans as well. And based on this screen, we selected the most effective drug as our lead compound. And then we continued by optimizing this drug, and also we started preparations of the first clinical trial of this drug, which started this year, and the first patient was dosed about a month ago, early March. So this drug is now called QR421A, and it targets a subgroup of patients suffering from genetic vision loss in Usher syndrome and non-syndromic retinitis pigmentosa. And the subgroup is affected by mutations within the USH2A gene, and then more specifically in a part of the USH2A gene, which is called exon 13. So our drug focuses, or is a possible treatment for all the mutations within this specific part of the USH2A gene. And our current estimation is that about 16,000 patients are affected by this mutation. So I would like to mention also that we developed this drug in collaboration with a foundation fighting blindness, and they support the the execution of the clinical trial financially, but they also provide a lot of knowledge about the know-how of the disease and also about the setup of a clinical trial design and, for example, the natural history of the disease, which helps a lot in the clinical research of this compound. So our first clinical trial is called Stellar, and it has just started. So the first patient has dosed. We aim to include in total 18 patients in this first trial to test the effect of the drug as well as the safety. This trial will be performed in 7 clinical centers in both North America as well as in Europe. So we have sites in the United States as well as in Canada and 2 sites in Europe. And we hope to be able to report data by the end of this year or next year.
I was diagnosed with RP when I was 12. And at that time, it was mostly my night vision that was affected. And I didn't want to get diagnosed because I was kind of a teenager and I knew my mom had RP and I just didn't care. Fast forward 15 years later, I started a PhD in biochemistry. And at that time, my daytime vision started getting worse. And so because I was in basic research, I had access to all the research articles and I started looking up on RP research as a hobby. And so finally, I decided to go to an ophthalmologist and I told him, I think I have RP and I think you should diagnose me. And so the process was, there was a lot of mixed emotions, and I'd like to highlight 2. And one was fear, and the other one was relief. So because of the diagnosis, it was clear that what I wanted to do, and there was a clear road path of what I want to do in research. So then I ditched that PhD, and then I joined Dr. Moritz's lab here in Vancouver. And so being in research, I dived into this vision research field. It was like, it was really amazing. I was like a child in a candy store and just soaking up all the information. And the meeting that's gonna happen starting tomorrow at Arvo, when I first went to this meeting, I was completely in awe and shocked. I have so much research going on. And then when I go to the lectures, the word lectures, I was always in tears at how much money and time and energy has been spent in this field. And so there's another thing I really love about going to this meeting is that there is so much swag at this meeting. And if you've ever been a student, you love swag. So there was one time, there was really this long lineup, and I was like, oh, this must be a really good swag. So I lined it up, and it turns out that there was an exhibitor showcasing this brand new machine that takes a picture of the back of your eye. So you've seen a couple of pictures from earlier today. They take the fundus photography, and what it turns out that it was an eye doctor operating this machine, and he was literally fortune telling people's lives by looking at the back of their retina. He was saying things like, oh, you must not sleep well, what kind of food you were not eating. And I thought, okay, what is this? But whatever, I want to get a picture of my eye so I can put it on my USB stick and put that into my PowerPoint presentation for the next science talk or maybe my PhD defense. And so he took a picture of my eyes, one each, and then he said, wait, I want to take another one. And so he took a second one, and then he said, do you have RP? And so he is a doctor. And I said, yeah, but can you tell me my fortune? And so he still hasn't told me my fortune yet, but I chatted with him for about an hour after that. And this is why I love about going to these meetings as a patient and as a researcher. And so if you're in the audience, or if you ever see another doctor, fortune teller, please tell him that I would like to know his name. Again, it's been incredible to witness how much progress has been made in vision research, even within my own lifetime. And the strength of basic research in academia is that there is freedom to be creative and innovative. And I'm going to steal a line from Dr. Bill this morning, who inspired me about basic research. And how many of you have heard about CRISPR? Can you say something? Because maybe I can't see you. Okay. CRISPR was discovered in basic research in bacteria. And this is why basic research is so important. And now I'm going to highlight some of the things that are going to happen in the next couple of days. And I just picked out very few that's going to happen at ARVO. So one of the studies done in cell culture, there is screening 69,989 small molecules in one single study to see if a misfolded protein in a cell can be cleared away. This is in one study, and it found 9 molecules. So I'm going to go check that out. In another study, also looking at small molecules, so drug therapy, they were able to restore the function. So not only it cleared away, it also restored the function of a misfolded protein in inherited retinal disease. And the third one, they found an FDA-approved drug, and this improves actual vision in a zebrafish model. The model is retinitis pigmentosa. And another thing that I think is really cool, they're going to spend an hour and a half dedicated to discuss combination therapy for inherited retinal diseases. So maybe you don't have to choose. What does this mean by combination therapy? Gene therapy, cell therapy, drug therapy, neuroprotective therapy, retinal prosthesis. Maybe you can pick 2 of them. It's like a shopping list. And for me, having hope for a treatment or a cure and believing in vision research is a wonderful thing. And this means that accepting vision loss doesn't mean that you can't have hope. And for those of you with vision loss, you should still go through the adjustment of vision loss. And if you're finding yourself having trouble to have vision loss as part of your identity, and here's where I'm going to put on my third hat, is I'm also an employee at CNIB, and my wonderful colleagues Sam and Carly are at the table with Vision Loss Rehabilitation Canada over there as an organization of CNIB, and they can help you through this process, but you can still have hope for treatment. Did you know that when you come to events like this in Vision Quest, you are the motivation and the inspiration for researchers? Because without you, there will be no one to do a treatment for. And because you're here, you remind researchers why they're doing what they're doing. So raise awareness, volunteer, and donate. And feel that how empowering it is to know that you are part of the fight against blindness. And this is going to be a part of history. Thank you.
I'm a basic researcher at the University of British Columbia. I have a lab over at Vancouver General Hospital site. And in my lab, we study the molecular mechanisms of inherited disorders of the retina. And sort of in a related, possibly basically the same thing, we study the cell biology of photoreceptor cells. So we're trying to figure out how they work and how they don't work under certain circumstances. And so primarily our research has involved studying retinitis pigmentosa, particularly the dominant forms of RP, but we've also done some work with cone rod dystrophy, LCA, Stargardt disease, and Noguchi disease. And principally what we do is we develop animal models of these genetic disorders. And so unlike our sort of twist, every lab has its kind of own twist on research, their own little niche. So our sort of niche is that unlike most labs which might study one or two animal models, we try and make as many different types of animal models as we can in order to compare them. So the motivation there is that if we understand the mechanism of a disease, we could potentially propose an intervention. But it's also important to know how many different mechanisms are present. So that if you come up with a therapy, you could then have some idea of how widely applicable it might be. So to facilitate this sort of idea of creating a lot of different animal models, we use a kind of unusual system, which is we use genetically modified tadpoles. So we make tadpoles with RP and tadpoles with LCA, and this is because these animals develop their eyes very quickly. So you have a little. Probably all remember from high school biology seeing pictures of fertilized frog eggs and how they develop into a tadpole. Well, that only takes about 4 or 5 days before they have an eye. And a lot of our tadpoles will have a RP-like condition by the time they're about 2 weeks old. And it's relatively much easier to genetically modify these animals than it is to genetically modify mammals like mice. So that allows us to do a lot of, study a lot of different genetic modifications in relatively short amounts of time. So just to sort of, I can tell you a story, I guess, which I was telling to some folks last night, but sort of give you an idea of what kind of comes out of this is one of the things that kind of inspired us early on is something kind of related to what Mike Sophia was talking about previously. There are a couple of research papers, case studies that were published in the 1990s showing retinas affected by RP, but the retina was not uniformly affected. So there's large areas of the retina that looked fine, and then large areas of the retina that were very severely affected. But that's odd for genetic disorder because all the cells in the retina have the same mutation. So why would some areas be affected and some not? It suggested that there was some kind of other influence on this problem besides the genetic modification or the genetic mutation. And so we made a couple of different animal models based on the mutations that cause this problem in these patients and looked at them. And what we figured out was that there was more than one reason for this. In fact, there were 2 reasons, and two very different reasons, two very different underlying mechanisms. And this actually resulted in the animals responding very differently to some therapeutic interventions that we propose. And these aren't, I would say, therapeutic interventions that are really maybe appropriate for human use, but sort of like a theoretical, what if we did this, what would happen? And the answer is something very different happened in the 2 different cases. So there's sort of an educational result that said, watch out. If you're going to propose a therapy, you really have to know what the underlying reason for the disorder, for the problem is. So that's just the sort of thing we look at in the lab, and I'll move it on to the next speaker.
Thanks to Fighting Blindness Canada for giving me the opportunity to talk to you a little bit about myself, but also about drug development. And I must say that the speakers this morning has made my job a lot easier because you've heard all about the different types of drug development that is actually in the pipeline. And I'll provide a little bit more detail as we start. So let's wait for the first slide. Now, Mary, you mentioned that the first meeting of this type was held in Winnipeg. So I'm very happy to hear that because I trained in Winnipeg. And so when I was trying to put this slide together, I just put together a couple of images that just explain my career and the 5 countries that I've lived or worked in. Canada, obviously, where I did my medical training, but I did my research in HIV in Kenya, in Nairobi. I first joined Bayer in the US, where I was part of the drug development team. And I've done a lot of work with our colleagues in Germany, because that's where a lot of our basic researchers are. And for those who are from Manitoba, you'll recognize the icon for the University of Manitoba, St. Boniface Hospital, the Health Sciences Center. That's where I worked. And this very colorful one is from the University of Nairobi. So I started in academic research, and it was HIV research. And this is where I first started seeing eye diseases, because back when I started treating AIDS patients, there was really no treatment. And one of the most dreaded complications we saw was cytomegalovirus retinitis, which is actually what that picture shows of the picture of the eye. And we had to work very closely with the retina experts, because really the best way to treat the more severe forms of that retinitis were to inject a drug called ganciclovir into the eye, or to give intravenous ganciclovir, which was quite toxic. And I mentioned this because I was doing this HIV research when Bayer found me, because Bayer was developing a drug for cytomegalovirus disease, which was less toxic. And I ended up joining Bayer and doing that drug development, at least to start with. But as I learned very quickly, drug development is very hard. So this slide, and I'll just describe it, describes a classic drug development. This is what we still do for many diseases, and I'll also explain how it's changing. But all of what you've been hearing this morning from the researchers, those are that's the research that gives us an idea of the mechanisms of diseases that still have need for new therapies. And our basic scientists go to work on that. And most big companies like Bayer, we have these very large libraries of molecules. We have one building basically where there are not a lot of people, but there's about 6 million different molecules. And there's a robot that goes around and can potentially screen each and every one of them against a target. And often for some diseases, you will screen 100,000 of these compounds to try to find just a few that may have activity. Of those, then you take them into preclinical studies, which are studies like some of the mice studies you saw this morning, to see if they actually do have activity against the disease of interest. And of those, maybe 1 or 2 will make it into the first in human study. And once you start those studies, the probability of success, once you're starting that study in humans, the probability that that drug will actually make it to Health Canada approval is only about 8%. So what I learned when I joined Bayer is that most of the time you're set up for disappointment. Virtually, the first 6 candidates for CMV I worked on all failed. And ultimately, we actually sold that research to another company. And about 15 years later, the first of the drugs from that class were approved. So that's a very long, drawn-out process. And it really is harder than rocket science, because, if you think about how much money and effort it took to put man on the moon, a single drug nowadays, like the ones you've heard about, like aflibercept, like bevacizumab when it was developed for cancer, the development costs can be over $1 million. Now, the Canadian dollar is a little bit weaker than the American dollar, so it may be closer to $2 billion. So that is actually more than what it cost to put the first man on the moon. Why does it cost so much? Now, I can give you an example from Bayer, because we have aflibercept. You may know it as Eylea. But for every indication that we develop it for, whether it's AMD or diabetic macular edema or central retinal vein occlusion, you have to do either a very large clinical trial or multiple clinical trials. And each of these actually cost a lot of money to do. There's some drugs we develop where a single clinical trial can cost $100 million or more. So you can see this adds up very, very quickly because not all of these studies necessarily succeed, but you need to do those to get that approval. So that's the very classic drug development. And it's these trials that make it so expensive. Now, we are involved in a number of different areas. So the research is changing. There was a time when companies did all of that research in-house, start to finish. Increasingly, these now are partnerships. We partner with the scientists that are working in universities. We partner with research organizations, and we partner with other companies to do this work. And aflibercept is a partnership between Bayer and Regeneron, for example. And through that, we try to find new treatments. And for eye diseases, we basically have those partnerships. We're looking at disruptive therapies. You've heard about some of those this morning, like gene therapy and stem cells, and I'll talk a little bit more about that. And then collaborating with other hospitals, other universities, and other companies. So two of the collaborations that we're really excited about. One is a company called Casivia. It's a joint venture, that uses CRISPR to target genetic diseases. And CRISPR is a way of editing human genes. So if you have a disease caused by a gene defect, sometimes there's only one letter in that genetic alphabet that is wrong. You can actually go in and repair it, at least in theory. And Casivia is active in clinical trials looking at these genetic diseases and whether you can cure them. So, previous therapies we developed treated chronic disease. The idea of these collaborations is actually to cure diseases, whether through gene therapy, whether through gene editing, or gene silencing, which is to actually use some of these technologies to actually perhaps shut down a gene that is overactive. The other collaboration we have is called Blue Rock, and I'm really proud of this because Blue Rock was the single biggest investment made in Canada, and it's in stem cell therapy to treat various chronic diseases, including neurological diseases, cardiac diseases, and eye diseases. And we are partnering with other institutions like Johns Hopkins for diseases such as Stargardt's disease, which is a genetic form of macular degeneration. The one other thing that we do, which is really important for all of the pharma companies, is that we have to work very closely with the experts, but we want to make sure that what we're doing is supported by the best science. So bringing experts together to develop clinical guidelines, to help us design our trials, to support awards that will help new researchers get started in the field, to start their first research. And then to support symposia and conferences so we can actually exchange ideas, to support preceptorships where new researchers or new physicians can work with established researchers to learn the science and to learn research techniques. And then finally, there are a lot of areas of unmet medical need, and I just wanted to touch on a couple of them because there's some really exciting research and technologies that are being developed. And I think the first and foremost, and you've seen a lot of pictures of the eye. The eye is one of the few places where you can actually look in and see the circulation and see the nervous system. So you can tell the future. When you look at someone's eye, you can get a pretty good idea of their health. And there are companies now that are using artificial intelligence and machine learning to look at those photographs and predict what type of disease that person may be at the early stages of developing, or will develop based just on the blood vessels in the retina, the eye. But more important, it allows us to very quickly screen for eye diseases. You know, when I was a physician looking at those HIV patients, the technology I had was the ophthalmoscope. You've probably seen physicians use it, and you could just see little pieces of the retina as you looked around the eye. And in my entire career, I might have looked at, 6,000, 7,000 eyes using this ophthalmoscope. But a computer algorithm can go through hundreds of thousands of images and very precisely identify the disease. So that is going to become more and more common because it's getting easier and easier to get those images of the eye. The other thing is, and the next things are actually sort of interrelated. We want to reduce the frequency of eye injections, and that has been mentioned, I think, by Dr. Merkur this morning. There are technologies coming that will help do that. There are new treatments that have a longer action, so you have to inject less frequently. There are port systems that are being developed, and if the phase 2 data are to be believed, you potentially only need to refill them once every year. So that greatly will reduce injection frequency. And we have also at Bayer looked at combination therapies. If one drug works, if you could look at 2 different mechanisms and block them, would that work better? Unfortunately, so far that approach hasn't worked out, but we have actually done a number of development programs where we looked at combination therapies where 2 drugs were injected in the eye, but they don't seem to be any better than just using aflibercept. Finally, there are technologies on the horizon where literally you're interfacing technology with the nervous system. And some of the, the early eye implants allows at least some vision to return. But there's a lot of research being done to build better interfaces that may allow, if you cannot use stem cells or gene therapy, you potentially could use actually a sensor much like the ones we have in our phone cameras now, which are quite small, to send a signal to the brain and send an image to the brain. So that's actually very exciting as well. So a lot of exciting things happening for the future across this field of drug development.
So one of the questions that I often get.
Okay. Going to stop there in the recording, and we're going to open it up now to your questions. And let me see. Okay, so I think we're back up. So yeah, if you have any questions, you can go ahead and put them in this box at the bottom of your screen. It's called the Q&A, and you can just type your questions right into there. Now, if you can't seem to access that, the other way you can ask questions is by what we call raising your hand. And to do that, you can use a keyboard shortcut. So on a PC, you press Alt-Y, and on a Mac, you press Option-Y. And that'll let me know that you want to speak. And then I can, when it's your turn, I'll open sort of the audio and you can ask through your device's microphone. So I have a couple questions that have kind of come in ahead of time. So we can start there and then go to the Q&A. So the first question for Dr. Moritz is, how do researchers decide on what treatments to work on or research?
That's a good question. Can you hear me? I just turned the microphone back on just to make sure everything's working.
Yeah, I can hear you. Perfect.
Okay. So in some cases, it's a question of history. So, I've tended to work for most of my career on retinitis pigmentosa, and I think a lot about it. And my research tends to continue to focus on retinitis pigmentosa. In other cases, there may be histories that are not specifically disease related, but maybe methodology related. So if you happen to be a researcher who's an expert in a specific technique, you may apply that technique to disease X, and then disease Y, and then disease Z. And then there's also a question of funding as well. You need money to do these experiments. And if money is provided in the form of a grant to do research X on disease X, and there's a lot of money out there to do research on that particular disease, it will tend to draw researchers towards it. So if at some point I ran out of money to do research on retinitis pigmentosa, I'd have to find something else to do, is what it amounts to. Did I answer the question?
I think so, yeah.
Okay.
That's good. Okay, so someone else was asking, can you comment on how failure, the failure of different therapies and experiments drives research forward?
Well, research, so failure is nothing, is 100% not unusual in research. You hear about the experiments that worked, but the experiments that don't work is the normal case, the normal state of affairs in the lab. So, I would say in my lab, you hit the correct answer about 25% of the time, I would say. You set something up, you go, I think I know what the answer is. I'm going to try this experiment to prove it. And about 25% of the time, maybe you're right. Probably about 75% of the time you're wrong, and you don't hear about those sort of things. But for the pharmaceutical industry, you see that all the time. Their failures are right out there because the clinical trial failed, right? And that does inform people on what to think about doing next, right? Usually, if the researchers are forthcoming, you can get some kind of clue as to what went wrong. Why didn't that work this time? Or just the fact that it didn't work. Just the fact that it didn't work. Sorry, just the fact that it didn't work will give you some information about what to try next. So yeah, I think that was alluded to in one of the speakers there, that there's sort of a very high failure rate, right? And it's just part of the fact that we don't know what's going on. You have to go down one path and then switch paths and go down another path, and eventually you get where you're going in the maze.
I think that's just a good life lesson in general, right? We learn from what doesn't work. So is there ways that the patient community can be involved in the development of drug therapies?
Sorry, can you repeat the question?
Absolutely. All right. How can patient communities or patients be involved in the development of different drug therapies? Is there any way they can be involved?
Well, one way for sure is to sign up for things like the patient registry for the Foundation Fighting Blindness, or sorry, the FBC, that allows people who are working on your particular disorder to contact you. And if there's a specific question that you could potentially be part of a study to help out with that study. There are other ways of getting connected to clinical studies as well. Depending on who your ophthalmologist is, if they're associated with a university, they may be involved in clinical trials themselves. And you can get involved that way. And then again, just by being part of the FBC community, by donating money, you're going to be helping out a wide variety of different studies.
That's great. Larissa, I don't know if you're there. Did you want to comment maybe a little bit on the patient registry and how people can get involved that way?
Sure, yeah. You can hear me, right? Perfect. Yep, so you can go onto our website and go to our patient registry page on the website, where there are instructions of how to get more information about the patient registry. You can also call our health information line. I don't have the number off the top of my head. I will find it and get back to you in a second, Morgan.
Perfect. And just in relation to that, Shaman asked in the Q&A, if they already registered for clinical trials last year, do they need to apply again? I'm not sure if they mean the patient registry or clinical trials, but can you shed some
So I
light?
can speak to the patient registry. So if you're already registered for the patient registry and have not only been in contact with somebody at Fighting Blindness Canada, but have actually talked to the intake site and filled in your form and got accepted by the registry itself, because there's a couple of steps in the process, then no, you do not have to register again. You're on file, you'll be on file for the foreseeable future. If you've signed up for a clinical trial in another capacity, I can't really speak to that. So if you've actually talked to a clinical trial that was starting or expressed interest in a specific clinical trial, you might have to contact them again to see what the status is of that. But that I can't tell you about that.
Okay, great. So we have some questions about sort of specific eye conditions, so we'll do our best. They may not be our area of expertise, but we'll try. There's actually a couple questions. I think Sherry was asking in relation, I think this was in e-news today, about vitamin D might not be great for AMD. And sort of if you have any sort of thoughts or opinions on that. She's been on it for 6 years, 200 milligrams a day, and wondering what this new research might affect her medication.
Yeah, so sorry, actually that's something I don't know very much about. I would sort of defer to the opinion of an ophthalmologist, of your own ophthalmologist on that. I'm not sure if that's a high dose or a standard dose of vitamin D. So I sort of be loath to comment on that particular question.
Yeah, and I think, Sherry, if you can also reach out to Health Info and we can give you more information about that research, and also obviously talk to your ophthalmologist, as Dr. Moritz said. Karen also asked about macular degeneration. She would like to know what supplements are recommended, any sort of supplements recommended for delaying or preventing macular degeneration.
Yeah, so there are specific formulations that your ophthalmologist can probably recommend a couple that are based on some studies going back a fair bit now. That the name of it escapes me, but the
AREDS, that one?
Yes, exactly. Sorry. The AREDS formulations of vitamins, and those are probably a very good thing to be on, actually, if you're if you either have macular degeneration or you've been your ophthalmologist has told you you're in danger of that, you know you have a lot of drusen and you look like you might be susceptible, but you're not actually diagnosed yet. Those are good things to be on that probably have very limited, they're probably going to generate a lot of problems for you. They're just, you know, a good vitamin supplement probably almost anybody could take. But they're specifically formulated for people at risk for macular degeneration or who have macular degeneration. And there's a couple of brand names out there, I think, that those come under.
Yeah.
I would just add something small. As Dr. Moritz mentioned, there are different formulations. So one of the formulations has beta carotene in it and one doesn't. And so with any supplements, if you're thinking of taking them, talk to your healthcare provider, of course, just to make sure that they do work for you. But one of them, the one with beta carotene, you should not be taking if you are a smoker, because there could be some negative impacts that way. So that would be just a little bit of a red flag. So again, another good reason to talk to your healthcare provider before starting a supplement.
Absolutely. Okay, so in the Q&A, we've got Leonard is asking, what is the biggest challenge for photoreceptors to connect or integrate in the retina, and how will this be overcome?
Okay, so that question is almost certainly referring to stem cell research, where people are growing, in fact, almost whole retinas at this point in Petri dishes, with the idea that you could potentially take somebody who's missing some cells in their retina, like photoreceptor cells, for example, and put them back. So grow them in a dish and inject them into the eye, and hey, vision comes back. But in practice, that doesn't work very well. you have an issue with getting those cells to, they don't just spontaneously hook up like they're supposed to. And so there are various different approaches being taken with this, but the underlying problem is that what you're doing when you put the stem cells back into an eye like that is something very different from what happens in nature. In nature, the retina assembles itself during development in a very specific order, and the cells start off as non-retina cells. They divide into various cell types and put themselves together to form a retina. And it happens in a very specific order and series of time. And so what you're saying is, hey, retina, here's, have some more of these cells. But they're coming at the wrong time and the wrong place. And the retina isn't waiting to have those cells, ready to have those cells at that time. So the question is, how do you make the retina sort of a fertile ground for seeding those new cells? And there's a lot of different approaches that are being taken to do that. But that's sort of, if you ask what's the problem, that kind of is the problem, is you're doing something that's not natural. You're not doing this at a time when the retina is ready for it. So there are certain things that people can have tried to do, like sort of trying to look at what are those sort of clocks that say I'm ready for these cells now. That can you sort of dial back the cell's sort of developmental stage a little bit and make them more fool them into thinking that they're a developing retina again. Or maybe it's just in some ways just a problem of certain barriers being in place, and you have to sort of distress the retina a little bit and open it up a little bit to allow cells in to take foothold. And there are various different approaches. Maybe you have to, even just dispersing the cells nicely over the retina is an issue. If you do an injection, things tend to just form a ball, and there are various approaches being taken to spread things out. But it's actually probably the fundamental, one of the most fundamental problems with the stem cell therapies right now, and they've been quite a few setbacks in that research in that regard. It's sort of the thing that bothers people the most, I would say, probably in stem cell research right now, is how do you get that to actually happen?
Sure. So I have some questions here about, someone said, are there any therapies being researched that may be helpful across the board for inherited retinal dystrophies, particularly where the genetic mutation is unknown? For example, I have a cone dystrophy for which the genetic type has not been found. Thinking about research, for example, that might be useful in halting cell death in a variety of dystrophies.
So yes, there is, that is a definite approach. And so you can broadly divide therapeutic approaches into a number of categories, and this would sort of fall under the category of inhibiting cell death. So if you have cells dying, there are various pathways that occur in the retina, or that occur in cells, that are sort of formal ways that cells die. And a lot of cell death have these pathways in common, regardless of the original sort of offending insult. So you may have mutation X, mutation Y, mutation Z, and they all cause cell death eventually by a similar common pathway. Pharmaceutical approaches that target these cell death pathways would be a sort of a general therapy. One of the issues with these general therapies, they tend to be a little bit on the toxic side, because inhibiting cell death is actually something that you don't want to do, because there's, or at least you don't want to do it sort of throughout your body, because there's a lot of natural cell death that occurs when, say, your immune system is targeting cancerous cells, for example. So things that inhibit cell death also tend to cause cancer. So you don't want to, you want to somehow be able to formulate that so that it's either less toxic or sort of retina specific. And that's where difficulties in that respect lie. And also you want to make sure that you're targeting the right cell death pathway. As sort of what I mentioned in my portion of the talk there. You want to be sure that the drug that you're using targets the cell death pathway that's actually occurring in the patient, so that you're not giving a probably somewhat toxic drug for a futile reason.
I just want to add in there, yeah, it was a really great explanation, Dr. Moritz. I just want to sort of do a little plug for one of our FBC funded researchers, Dr. Kathy Tlsvidis from the University of Ottawa.
Yeah, that's an example right there. Yeah.
That's exactly what she's doing, is trying to do a gene therapy that is inhibiting cell death. So
yeah, and that would be, you know, it might not be applicable to all retinal disorders, but the idea would be it would not be applicable to only one, which is, you know, a gene therapy where you replace the gene of interest that causes LCA or what have you. It's only going to work on that particular form of LCA, right? But something like Dr. Tlsvidis' research might be applicable to a large subset of retinal degenerations.
Yeah, and Dr. Tlsvidis, she spoke in Ottawa earlier this year at our Vision Quest before COVID kind of took over, and she did a great talk, and it's actually on our YouTube channel. So if people are interested in learning more about her research, she has a whole talk up on our research or on our YouTube channel as well about her research. So I'm a bit mindful of time. I know we have a few more questions, so I'm going to ask, I think, maybe two more, and then we will stop. So someone was asking, I live with retinitis pigmentosa, and my sibling has Stargardt's. How does that work?
Okay, so a good question would be, two siblings have two different diagnoses. Yeah. Is that correct?
Yeah, RP and Stargardt's.
Okay, so Stargardt's is an interesting one because there's only a few causes of Stargardt disease, and there's one particular one that causes a lot of Stargardt disease, which is mutations in ABCA4. And some people are of the opinion that only ABCA4 causes Stargardt disease. So an interesting question would be whether you have a genetic diagnosis for you and your sibling and you have the same genetic problem. So my guess is you probably do, and that either you have I can't remember who has the Stargardt and who has the RP, but the RP and Stargardt diagnosis by symptoms is kind of being displaced towards diagnosis by genetic disorder. So if you would say, okay, well, we both have mutations in the ABCA4 gene, well, you probably really both have the same symptoms, or sorry, the same underlying cause, but your symptoms are manifesting themselves a little bit differently in the way your retina looks. And the ophthalmologist is calling it RP in one case and Stargardt disease in another. And that may be just a bit about the geometry of how the disease is progressing. But that could be, you know, you have a genetic influence on your disease. You also have, that's, you know, the direct cause by the mutation that's the principal cause. But there are other possible moderating factors that could change the way the disease develops. For example, you're not eating the same food as your sibling. You have different the other genes in your body are different, and the environment that you live in is different, right? So there's other possible things that could change the way the disease develops over time. And so you may have a slightly different appearance to your retina, which could possibly result in two different diagnoses. The other possibility is you actually do have two different genetic causes, but that actually seems quite unlikely to me. Probably not.
Great, thank you. That's interesting. So we have a bunch more questions. We're going to just do one more, and then I'm sorry if we didn't get to your question. Either myself or a health information officer will reach out to you to try to get you some answers. But last question, Ivy is wondering if there's any results or research on gene therapy in Stargardt's disease with gene editing with CRISPR or Cas9 technology that you know of.
So I'm going to say almost certainly, but I don't know of any specific results related to Stargardt's disease and CRISPR-Cas9. I know that there are, so the Stargardt disease treatment, this is specific to caused by the ABCA4 gene, I'm not sure. But as I mentioned, most Stargardt's disease is caused by the ABCA4 gene, and gene therapy for ABCA4 is complicated by the fact that the gene is really, really big. And so it's difficult to fit into what they call the vectors that people use for gene therapies, like the gene therapy for RP65 LCA that was quite successable. So researchers are trying to figure out ways to fit these, either make the vectors have more capacity or shrink down what's necessary to put into these vectors for gene therapy purposes. And as far as CRISPR-Cas9 based research, I guarantee there's something going on. I don't actually, off the top of my head, know what that would be. I'll say right about now I would be typically at my most informed because as we saw in the previous video, the ARVO meeting, which was in Vancouver last year, is going on. And normally like I get to go to ARVO and see all the absolute latest stuff that everybody's doing. And unfortunately that didn't happen this year because of the pandemic. But yeah, you know, the CRISPR-Cas9 has a lot of potential to do similar things where you could put in a sequence that's incorrect. You wouldn't have to put in necessarily even the whole gene if you use CRISPR-Cas9, but it's susceptible to the same issues, which is that the CRISPR reagents themselves that you have to get in there are very large. And the gene therapies involving CRISPR are difficult to construct in that way, in that they the sort of standard methods that people have been using for developing gene therapies for a while aren't quite as applicable because the things that we need to get in there are so big. So again, you know, the research just on figuring out how to get CRISPR in, regardless, would be sort of appropriate to ABCA4 research, even if it's not specifically about ABCA4.
Thank you. I think we're going to unfortunately have to stop there. But as I said, if you asked a question and we haven't got to, we'll reach out to you. So thank you so much, Dr. Moritz, for joining us. And we're really grateful that you were here to answer our questions and to share that discussion about the new sort of what's coming up in pharmaceutical therapy.
Okay.
Thank you.
Well,
So,
thanks for having me.